Okay, hi everyone. We are now finally ready for me to start. So this afternoon we have a bit of a mixed lecture. You're going to have me for roughly half of it, and then you'll have John taking over. And we are going to be talking about statistics, visualization, and finding functional significance in our data. So first of all, is anyone else feeling a little bit like this by the end of our show of hands? Anyone feeling like this, or are you feeling better? Anyone feeling better than that? No, no one. Okay. Oh, we had one. Okay, we had a couple feeling better than that. Great. So as I'm sure you've kind of all been learning in this slightly crash course to bioinformatics, bioinformatics is not always straightforward. There are many things we can do at many different times, and often we're going to be feeling a little bit like this, overwhelmed, tired. There are a lot of different things we can do, but hopefully at the end of it, we kind of come through. And for this lady in the picture, she was the first person ever to finish the Barkley ultramarathons last year. And then she went on to become, she got awarded, I think, British Sportsperson of the Year after that. So this was quite a feat. This race has been going for over 30 years, and I think about 25 people have finished ever. Bioinformatics hopefully is not quite that bad. Hopefully a few more of you can finish than that, slightly better odds. But it definitely can be feeling a little intense at times. So in this part of the lecture, hopefully you're going to be learning a little bit about how we use stats in microbiome analyses and how we incorporate some confounding variables into statistical analyses of microbiome data. So before I begin with that, I just want to give you a little disclaimer that I'm going to be talking about what we often end up doing in practice, but I am not a statistician. So we're often, as biologists, I think, a little bit scared to give advice on stats because often statisticians kind of come along and tell you, like, you're wrong, this is what you need to do. They're quite black and white about things frequently. And I think biology is often a little bit more gray than that. You know, there are things that would be great if we could do, but we're not always able to do that with our data. And so with that, if you think I'm wrong, yeah, I probably am. I'm okay with that. You can tell me I'm wrong. Yeah, I'm going to be talking about what we often end up doing and some of the ways that we kind of try and get around some of the oftentimes many confounding variables in our data. But this is not perfect. There are different ways to do it. There are always different methods coming out as well. We often try and give some of those new methods a try, but that doesn't mean that they're always going to be the right thing to do. So yeah, speak to a real statistician if you're concerned about any of this. So with that, I stole these slides from last year. Just as a brief overview, in statistical inference and hypothesis testing, what we're usually trying to do is understand a population level system from a sample of data. And for this, we often use null and alternative hypotheses. And along with these, we have test statistics and p-values that are essentially supposed to tell us, do we accept or reject the null hypothesis? But statistics is a rapidly evolving field, and data analysis often feels a little more like an art than a science. And just before I go into all of my kind of hand-wavy part of this, where I just tell you that it all depends, which seems to be the answer we've given to most things over the last few days. There are many different types of statistical tests that may or may not be appropriate for your data. This is just kind of an overview that you can look at another time if you like, but just to demonstrate that there are many different statistical tests that you're going to need to work out if they are appropriate for your data or not usually. And there are also many problems with hypothesis testing. So many of you have probably heard of the idea of p-hacking and misinterpretation of p-values. And so if you look this up on Google, this tells you that p-hacking is the inappropriate manipulation of data analysis to enable a favored result to be presented as statistically significant. And the way that you'll usually see this is in people just continually running tests until they find a result that they like. And so for some of the beginner microbiome workshop, when we talked about differential abundance a little more, we mentioned that what we often like to do is run a few different tests and present the results of each of those And then whoever is reading our paper can make a decision for themselves as to how significant they think our findings are. We don't want to just run a bunch of different tests and then present the results of one of them because they were what we liked the most. And ultimately, using different tests does affect the conclusions that you're going to come to. And so it's nice to often present a variety of those, and we particularly like it if the results of each of those tests overlap with one another. That gives us a lot of confidence that even if we did something slightly different with our analysis, we'd still be coming to the same biological conclusions. And so there are other kind of alternatives and complements. You might be looking at data visualization. You always want to visualize your data because just seeing that something is statistically significant doesn't really mean anything if you have a very tiny difference between two groups, for example. So then you're also looking at confidence intervals, effect sizes, comparisons of different models. But hypothesis testing is obviously still a widely used method, and it's a kind of systematic way of evaluating scientific findings. So now jumping slightly to sequencing depth, which John did kind of talk about a bit earlier. One of the things that we're usually needing to control for is varying sample depths or sequencing depths, which we kind of use interchangeably usually. And so this kind of refers to the idea that the sequencing depth is not directly related to the original amount of cells or DNA. And so more depth doesn't mean more microbes, but greater sequencing depth does mean that we have a greater ability to detect different taxa or different functions. And we therefore typically need to normalize or control for sequencing depth. And so in amplicon sequencing, we have methods to come around this. We often use rarefaction, conversion to relative abundance, conversion to log ratios, and we sometimes want to take into account gene copy number variations. But when we're looking at metagenomic or metatranscriptomic data, we're often wanting to normalize for gene or genome length as well. So essentially, as John said, if either a gene or a genome is bigger, then there's a higher chance that they're going to be sequenced. And so we're often looking for the reads per kilobase per million, possibly. And the way that we often use this in metagenomics is we don't always want to normalize to that in the same way as the per million. Sometimes we might just be including the read depth as kind of a confounder in our statistical tests. And so then that brings us to kind of what else we should be including in our statistical tests. And really that's any other confounding variables. So any variable outside of what we're specifically interested in our study. So for human studies, this could be things like gender, age, ethnicity, diet, socioeconomic factors, whole host of different things that we might be wanting to include. And essentially, we're often looking to include just as much information as we possibly can gather about these populations. And it's the same for environmental studies, that, this could be depth, light availability, nutrient availability. All of these things are going to have an effect outside of probably just the variable that you're actually wanting to look at in your data. And so I want to just give a couple of examples of how this might be important. So looking at this kind of question, like, does geographic location impact the microbes found in seawater? So I'm focusing on one of the Tara Oceans study. So for anyone unfamiliar with Tara, essentially this was a massive sampling effort undertaken a few years back. They went on cruises all around the world, collected samples from different oceans, different locations, different depths, different size fractions of the seawater they were sampling. And essentially, if you were looking at, does geographic location impact the microbial community, you may well find that, yes, it does. But actually, what seems to be more important in something like this is the depth. And so if they hadn't included depth as a factor, then they wouldn't know from looking at some of the rest that it's actually way more important in this case than just the location of these samples. So what this is kind of showing is how we have, all of these different regions that these samples are taken from, but it's actually the depth. So that's shown in here with the different shapes and then plotted out on the bottom, that is explaining most of the variation between these different microbial communities. And so then I want to give an example from some of our own work about how we kind of approach finding out what the confounding variables are when we're carrying out a microbiome study. And so I don't really want to show you any results here, but I want to give you an idea of kind of how we go through this and think about the kinds of things that we're going to want to include in our statistical models. So the data that I'm talking about is from a project where we are looking to see whether there's an association between the oral microbiome and mental health in children. And so this was collected from children that attended the Ontario Science Centre. About 22,000 were enrolled, and they were asked quite a long questionnaire and also to give a sample. And so this is called the Spit for Science project because they were literally asked to spit for science. And originally these were collected so that they could do human genome sequencing, but we've then been able to use these same samples for the oral microbiome. And so of these approximately 22,000 participants that were enrolled, we used a subset of just over 5,000 that already had their DNA extracted. And at the end of all of our quality filtering steps, we had close to 5,000 samples. And so if our question is looking at whether there's an association between the oral microbiome and mental health in children, then there are quite a few different things that we're going to want to consider. So some conditions are more or less prevalent in older children. So I've given the example of bipolar. This doesn't tend to be diagnosed in young children. It tends to only come out when they're older teenagers or perhaps even older than that. And so then if we want to have a look at this, then we also need to know, how does the oral microbiome shift with age in children? What are the differences going to be that we're seeing due to age, rather than just, if we just looked at whether they have bipolar or they don't, then we're going to be ignoring that kind of confounding factor of age. And it's the same that some conditions, for example, ADHD, are more commonly diagnosed in male children. I mean, obviously some others could be more commonly diagnosed in females. And so then if we want to start digging into that, then we need to know how the oral microbiome shifts with gender in children. And then again, we have, okay, so the slideshow doesn't show. There was some animations here. It wasn't always covering it up. But there are other conditions that are going to be more commonly diagnosed in white children than children of other ethnicities. That's not because there's anything different necessarily about white children. It's about their access to healthcare and how frequently they're seen, cultural differences, whole host of other factors. But then if we want to know about that, again, we need to know how the oral microbiome changes in children of different ethnicities. So again, this looked different before. But then we also want to know about what other factors there are that impact the oral microbiome in children and also might impact their symptoms that are associated with a mental health condition or their likelihood of a diagnosis. And so some of these other factors might be things like socioeconomic status. Are they more likely to have a parent that's able to take them to a doctor and receive a diagnosis for something? You also have their diet. This can be I guess, both impacted by and impact some conditions, and their environment that they're living in as well, their access to green space, their access to kind of parks and recreation facilities. So these are all things that we might need to be taking into account. But as I'm sure you can kind of imagine, there's a lot of different factors here, and that can be really complicated to try and add into something. And so it kind of brings us on to how we might account for multiple confounders. And so the simplest way of doing this is just to include all of these other variables in our statistical models. So the way this looks like when you're kind of writing an equation that's going to go into your stats test is that you're going to be looking at your data and how it associates with your main variable plus your confounding variable, another confounding variable, and so on. And so for this example that I've given, this is going to look like, kind of including your data, looking at age, looking at gender, looking at ethnicity, looking at diet, and so on. But this can be kind of problematic because trying to include tens or even hundreds of different confounders really increases the burden of multiple test correction that we're trying to do. And so that's kind of what we're aiming to do to control the false positive rate here. Because essentially, if we run more tests, then we're going to end up with more significant results. But that doesn't mean that they are kind of biologically relevant, because the more tests that you run, the more likely you are to have a significant result kind of by chance. So usually we do multiple test correction, but if we're ending up with thousands of tests across thousands of different taxa, then this is going to be really complicated and we end up with nothing that's significant at all. So what we often end up doing is a couple of different things that so what we might do is we might have two different models for something. So we might test the confounding variables individually. And And like most of biology, there's not really a single perfect approach that we can use. And so if we're looking at, say, using a couple of different models, this is an example that we did recently where we looked at people with IBD and people without IBD. And essentially this first statistical model, we were just looking at cases versus controls. For these participants, not every single one of them had every single metadata variable. And so then we reduced the numbers of participants to just those that had all of the available metadata. And yep, we saw that there's still a significant difference. And then the final way was that we did the cases versus controls with all of our covariates. And so in this case, this was still a significant stats result. There was still slightly lower diversity in cases versus controls. But in many cases, we lose significance once we've accounted for all of the other confounding variables. And so we weren't really sure what we should do with this. So we just thought, if we just present this, then at least anyone that's looking at it can kind of see all of the information there, and they can make a decision for themselves as to whether they think it's kind of a significant result that you have this reduction in diversity. So then the other approach is the kind of testing the confounding variables individually. And that's a bit more of what we've done with that Spit for Science cohort that I mentioned. So here we were testing each of these variables in both a univariate, so that means you're just looking at one of the variables at a time, and a multivariate model. And we were looking to see whether it's still significant when we're including other variables. And so you'll see that for something like this, this means it's a healthy eating food index. It was significant when it's looked at on its own, but not when you include all of the other variables as well. And so then that kind of brings us on to another way that we might try and reduce the number of confounders. We might be interested in whether there are summaries of variables that we can use that will explain most of the variation. And so for something like diet, we have a bunch of different questions where we have information on, how many grains they're eating, how often they're eating fruits and vegetables. But is there something that we can summarize from that that will be easier than us including 25 different diet questions in our stats tests? And so we thought that maybe we could summarize this with something like the healthy eating index that I just showed you on the previous slide. Or maybe it's just the number of different things that they're eating that matters. Is it just kind of a variety? But in this case, no, these were all of the different kind of categories of food that we were looking at. And this is that summary variable in here. So we actually find that including each of these variables on their own is much more effective for looking at the variation that they're contributing to within the oral microbiome than just including a summary metric. However, in other cases, if we look at something like the deprivation index, so this is kind of developed by some people to look at the Canadian census data, and it's been calculated from, I think, about 6 different questions within that, and they've actually found that they can reduce that just to 2 principal components that explain the majority of the variation. So in that case, we're able to include that instead of these 6 different components. We've reduced it down to 2. And then finally, can we group some of our variables of interest? So in some cases, we have things like cases versus controls that we can run tests for. And so for something like having a bacterial infection or not, we may be able to just kind of categorize people into cases versus controls based on a certain bacterial load. And so maybe there's kind of a tipping point above which infection occurs. So just in this kind of basic example, maybe you've got kind of 3 bacteria and you're uninfected, and then you have 4 and that's an infection. Most things in biology are not that simple. And so for when we're looking at the mental health in these children, there are quantitative traits associated with many of the mental health disorders, and these are things that they use to give a child a diagnosis. So for something like ADHD, it's called the SWAN scale, that they're looking at to see whether they're, likely to have traits associated with it. And in the clinic, you might say that someone has a diagnosis when they're above the kind of 95th percentile on the scale or something like that. But if you're looking at associations with different taxa, it doesn't really seem particularly likely that above this 95th percentile, you just suddenly have this appear. And so in that case, does it really make sense to look at this kind of diagnosis versus no diagnosis, like cases versus controls, when you actually have a continuous scale for something? And so I think it's really important to think about, if you're looking at grouping variables, it's important to think about whether that biologically makes sense. whether they should be treated as categorical or not. And so just finally, I want to mention power calculations, but I don't know how to do them. So according to Google, a power calculation is a calculation done before a study begins to ensure that it has a reasonable chance of finding a true effect if one exists. And essentially, this is looking at whether we're collecting enough samples to see an effect. Many grants will ask for these, and there are articles published that tell you about how to do them. But I'm going to refer you to these modules from one of the previous years that we ran this workshop with CBW and also Impact. They're called. They had a couple of different lectures, and these links within the slides go to the YouTube videos of those lectures. So if power calculations are something that you are interested in, then please explore these lectures. So with that, are there any questions on this part before I hand over to John?
All righty, so I am going to spend the rest of this 30 minutes I've got talking about some additional interpretation, visualization of these kinds of data sets. And I want to focus on, again, this is something that was touched on during the beginners workshop, which is these tools to identify significance in our data sets, whether it's changes in abundance of taxa, whether it's changes in abundance of genes in metagenomics data sets, or whether it's changes in the expression of genes as well. So how do we go about identifying these significantly differential features, whether it's taxa, genes, or the expression of genes? I guess the TLDR version is that, particularly for metatranscriptomics, I think also for metagenomics, the statistical tools that we use for identifying differentially abundant or differentially expressed genes don't work. I don't think we have any tools that are very good at identifying these kinds of genes. And so we can make a pretext of using something like DE-seq2, and I'm going to go over DE-seq2 to try and identify these genes, but I'll explain why I don't think they do a great job. But I think the power really comes into these gene set enrichment analyses, which is getting back to this idea of looking at these submodules, these functions, right? If we can see sets of genes that have this coordinated pattern of changes in abundance or changes in expression, that those kind of ideas, that enrichment in specific functions that are upregulated or downregulated, that's giving us potentially the statistical power to actually give us some information that we feel confident about. And again, this is about hypothesis generation. You want to go back and you actually want to validate some of these effects as well in more mechanistic studies. So a lot of this kind of where we're trying to find significance in our results, yes, we can try these methods and you can run them, you'll end up with results. Reviewers probably won't question them. But it is, I think the power is really in collapsing sets of genes into these gene sets so that you can show that these particular functions, this particular pathway, this particular protein complex, all the components seem to be changing together. And I think that's where the real power comes in and becomes more convincing. I also want to introduce this concept of topic modeling. Has anyone heard of topic modeling? Yes, Tony. Anyone else? Okay, so I think back in about 2010, 2012, there were these studies that were coming out, some of the earliest studies of human gut microbiomes. And there was this suggestion that you could partition individuals' gut microbiomes into specific enterotypes. They're called enterotypes. And so we all had this enterotype, and I don't know if there was 10 or 20 of these enterotypes that were three. There was three original enterotypes, right? So your gut microbiome is one of three enterotypes. And I think over time, it became fairly obvious that this is a little bit of a rubbish idea, but maybe there's some nuance to this. And in fact, maybe there are sub-communities that are groups of bacteria that are coming together as individual sub-communities and contributing to your larger community. So these are groups, these are almost like these cliques that I think Ryan was presenting in these networks. Topic modeling is another way of kind of identifying these kind of cliques, these kind of groups of microbes that tend to travel together, tend to be present together in the same microbiomes. And it just gets, maybe it's a lot more nuanced. It's like there aren't enterotypes, but there are certainly groups of taxa that tend to be coming down together. And this kind of topic modeling seems to be a fun way of being able to identify these kind of cliques. And then finally, I just want to touch on visualization of microbiome data sets. I think this is something that I struggle with in terms of coming up with visualizations that are intuitive and enable you to explore and interpret the data in a meaningful way. And I don't think, again, as a community, we're doing a very good job of kind of visualizing the kind of data after we've done the processing. So the ANVIO, for example, that's really nice diagram. It's kind of intuitive. It shows you sections of genomes, for example, which are shared across different strains. And so you can get this concept of, I guess, lateral gene transfer, or even where different lineages may have mixed if there's been some kind of recombination events. So you can look at these kind of events and there's a nice visual for it, but it's pretty specific. So I'm wondering if there's, again, room for creating tools that enable us to get a more intuitive sense or an intuitive way of looking at these data sets. All right, but first of all, I want to start on these differentially abundant genes. Okay, so this is what we're tasked with, right? This is our problem. We have this big matrix. After we have generated, whether it's your metagenome analysis, whether it's a metatranscriptomic analysis, you have a bunch of samples, you have a list of genes. In this case, this is from a poultry gut microbiome, 184,000 transcripts across 73 samples. So how do we interpret these data sets? How do we find some kind of meaning in these data sets? And so the approach that we typically do is, first of all, can we identify the genes that are exhibiting some kind of significant difference in abundance or expression? Abundance if it's a metagenomics, expression if it's kind of a metatranscriptomics data set. Okay, so you want to come up with a list of genes out of that 184,000 transcripts, which look as though there's difference between those samples versus another samples. Those are the genes that are driving those differences. And then the second step is to see if these genes are then enriched in specific functional categories, right? So you could use DESeq or any of these other differentially abundant tools to identify your list of genes that you're going to feed into these gene set enrichment analyses, or you could simply just use fold change. And there's nothing wrong with saying all of these genes that are, say, one log fold different between this sample versus that sample, are they enriched in specific functions? Are there functions in there that these genes are contributing to? And again, that gives you a nice kind of statistical test to explore without having to identify if these genes are significant or not. So why am I so down on this kind of differential abundance? So I believe Morgan introduced this in the beginners workshop. So this will be the next couple of slides will be familiar to those of you in the first workshop. So there's multiple tools that have been developed to identify differential abundance or expression of genes. These have largely been developed with RNA-seq data sets in mind, right? And they're not appropriate. They're not really appropriate for use on metagenomics or metatranscriptomics data, which our matrices are much more sparse. We have way more transcripts and genes relative to a standard RNA-seq data set, which looks at one organism. And we might have more samples. And so it's a really zero inflated data set. And a lot of these statistical tools are really badly designed for looking at these zero inflated data sets. So the two I want to call out here are DESeq2, which is one that my group has used and adopted for multiple studies. It gives you a lot of hits. So we like that because it gives you a lot of hits. There's a lot of false positives, but it gives you a lot of hits. There's another tool that we've started leaning a bit more on. This is Ancom BC. Ancom BC, and there's another one, Aldex2, which you can be pretty sure that if they find a significant gene, they really are significant, but they don't give you very many, right? So across some of these data sets, these are individual data sets. These are looking at the performance of different statistical tools to identify significantly differential abundant, I think taxa are in this case across these different data sets. And we'll see that typically for a data set, if DESeq will give us something like 1,000 differentially expressed genes, Ancom or Aldex will give us about 20, right? And so I have a colleague, statistician, we sit down, we talk about, how do we identify statistical significance? He says, well, you don't have enough N to do this. He says, you could use this tool, but then you end up not finding anything. And it's kind of like, well, Andrew, we've generated this data. We need to do something with it. So can it not be quite so negative? So we're using these kind of tools just as a way of pruning the list of things to focus on in the first place. But really, again, to emphasize, I think the power comes in with these gene set enrichment analyses. So Ancom BC, we like it. This paper that Morgan's group did shows that Ancom BC actually has a greatest overlap with all the other tools as well. So it's fairly consistent. So if you are interested in really identifying genes that are really super significant in your data set, then Ancom BC is probably your tool. I wonder if part of the problem also with the use of these tools is that our N is too low and we're really needing fairly consistent data sets with very few variables, i.e. not having different diets, not coming from different geographies, not having different genetics, and you need an N of 6. So your mouse studies need your N of 6, for example, in order to start getting more reasonable statistics. Robyn.
Ancom has a newer version lately that is quite different. I don't know what's different about it, but the results it gives you are quite different. So it's worth bearing in mind that the most recent version of it is probably going to be quite different places around here.
Okay.
I don't know if that's good or bad.
Okay. All right. Thank you. Another complication we have with metatranscriptomics is, again, we don't have any good tools for identifying differentially expressed genes in metatranscriptomics datasets. And there's this complication that if you have a shift in abundance of your taxon, that's going to contribute to you seeing a shift in expression of genes, because you've got more of that taxon, you're going to get more expression of all the genes associated with that taxon. But in that sample where you get more of that taxon, you're going to get higher expression of those genes as well. And so there's this concept of taxon specific scaling. So we have these kind of 5 different cases here where we have a baseline, which is the real data. So the blue is metagenomics, the red is metatranscriptomics data sets. And so under case 1, what happens is if you see an increase in genome B, you're going to get an increase in the metatranscriptomic profiles simply because you have an increase in genome B, for example. And the real case is in class 4 there, where you actually have a change in expression of gene 4 in taxon B, and that's really because there is a change in expression. So there are these different things that can contribute to these kind of signals where you might see an elevation in expression or whatever, and this is due to taxon changes. Okay, so if your taxon changes, that's really going to alter what you're going to see in terms of expression.
Oh, that is easy. It's just literally like visualizing it there or dividing by your number of text. No other.
Okay, so yes, Yes. So potentially what you could do is if you have a metagenomics data set at the same time as a meta-transcriptomics data set, then that's great. You can do this taxon-specific scaling, and then you can do that normalization based on what the baseline of the mags are or the metagenomics is, right? So if you have that, then absolutely you can normalize it on that basis. A lot of us don't generate metagenomics and metatranscriptomics data sets at the same time simply because it's expensive, right? You're almost doubling the cost of the experiment. And so is it better to do more samples, or you're looking very puzzled there, Morgan? No? Okay, all right. So again, it comes down to your cost and your funding, and is it better to do more samples, or is it better to do more comprehensive characterization on more limited samples? So one thing that we did is this thing called log-centered ratios. So this is where we've got 3 different pathways here. So cell wall biogenesis, glycolysis, purine metabolism. And what we're doing is we're comparing the expression of each of these pathways in these different taxa. So each dot represents an individual taxon. And then we're comparing it with the overall expression with that taxon. And in this case, we can perform these tests that we can see that these pathways are exhibiting significant differential abundance compared with the overall expression of all the genes. Okay, so this is one context where we can actually use the global gene expression as your kind of baseline. And then for your specific pathway, are all the genes associated with that pathway, are they all significantly higher, or are they significantly deviating in expression or abundance relative to that kind of baseline? And so this gets at this concept of this taxon-specific scaling. So if you do have a sense of what the actual abundance of each of your taxon is, then you should be scaling your expression associated with that taxon or your abundance of that gene associated with that taxon based on the relative abundance of that taxon. Okay, so this is still in development. They tried, I think, 6 different models, this from Curtis Huttenhauer's group. And at the end, it was all, yeah, again, I think the take-home message from this study was you need to do metagenomics at the same time as metatranscriptomics. And so from a practical point of view, what can we do? Again, probably the center log kind of normalization is a way forward here to see how much your specific groups of genes are being are in higher abundance or in higher expression relative to the background level. So any comments on that? No? Okay. All right. So just to show you why I'm so skeptical about these approaches. So this is from a data set. It's just 30 infant gut microbiomes. And these were infants that I think half of them, 15 of them, were born to mothers with gestational diabetes. And the other half were from mothers that didn't have gestational diabetes. And so we were looking at 4 different factors here, whether the mother had gestational diabetes, whether there's a difference in mode of delivery, so C-section versus vaginal delivery, the sex of the child, and then exclusive breastfeeding or not, right? And so we applied taxon-specific scaling, that's the bars in orange, and then we also used the seq using the pos counts and the base kind of difference as well. And the thing I want to highlight here is those sort of 3 bars on the left-hand side. That is what happens if you take your data set and you just scramble up all the labels. That's how many genes you will predict as being significantly differential abundance using each of those 3 approaches. So this suggests that your DE-seq2 is giving you nothing better than noise, right? You're getting the same numbers of genes as this kind of random data set. And this was kind of somewhat disappointing to see that. What are you actually getting from running these kinds of tools, DE-seq2? Okay, so this is a problem. We can generate these individual lists of genes, whether it's through DE-seq2, whether you believe whether they're really significant or not. Or you could use fold change as another criteria, but you want to get that initial set of genes, that initial list of genes that you think is potentially enriched in something. And once you get that initial set of genes, then you can start thinking about doing these gene set enrichment analyses. So you can, for example, use gene ontology categories. And what you do in these gene ontology categories is you're trying to identify, are there certain functions associated with the groups of genes in your list that are enriched? So how does gene set enrichment analysis work? You start off with your list of genes. You then choose your gene set. So this could be gene ontology categories. It could be these KEGG pathways or modules, although this seems to be problematic now that they're charging $10,000, was it, Robyn? Okay, which again is really depressing in terms of the lack of resources that we have for these kind of functional categories and functional modules. There's a seed database, which I guess we haven't really touched on the seed database, have we? So the seed database, this was a project that's associated with MGRAS. There's another tool that uses the seed database. This was coming out of, it's a Midwest consortium, and they had something like a $50 million grant to produce these kind of resources for studying bacteria. And so the seed database was their way of coming up with some kind of ontology. It's really, really clunky, and we don't really use it or promote people using it because it just, again, seems not very intuitive, but it is there if you want to use it. There's COG categories, which is just a limited set of, think of 20 or so categories of functions, which has remained relatively untouched since 2003, which doesn't seem very good. Or you could have your curated subsystems. So these could be genes involved in, for example, ion transport. So you could use all of those as a set. Okay, is your set of enriched genes or abundant genes or these differential genes that you've identified in your first step in your list of genes, are they enriched in genes associated with, for example, ion functions, or are they enriched in things to do with AMR? And so that's another possibility as well, rather than relying on these very broad categories. And then there's a number of methods out there. Cluster Profiler, the one that we use is FGCEA. This is an R script. It's very easy to run. It just reads in your list of genes. It reads in your gene set, and then it assigns the probabilities and identifies within the gene sets which ones are functionally enriched, which ones are enriched in that list of genes that you're giving it. So just to give you some examples of how we've applied this under, again, back to this Plin2 knockout mouse, we use these gene sets that consist of KEGG pathways to identify specific pathways that were enriched in genes that were differentially expressed. And that pointed us to certain energy pathways, which, as I mentioned this morning, may suggest a response of the microbiome to an elevated high-fat diet. We've looked at poultry gut microbiomes. We've used gene sets based on AMR genes, and we've done these statistical tests to identify significant groups of antimicrobial resistance genes that are elevated in response to the addition of these antibiotic growth promotants. And then I also mentioned this morning that when you colonize these mouse guts with this protist, Tritrichomonas musculus, again, we use these iron-related genes as a gene set to show that there is enrichment in some of these significantly differentially expressed genes associated with these iron-related pathways. So this can be a very powerful approach to actually identify whether your function is significant or not. Sometimes, depending on if you're focused on more of these broad categories, it can be challenging to interpret what the data is actually telling you. So you're getting these very broad carbon metabolism, for example. What does that mean? So there's a difference in carbon metabolism. What am I supposed to get out of that? What kind of mechanistic insights can I get out of that? And so if you're looking at gene ontology categories, gene ontology is this hierarchy. And so there are subnodes in this kind of hierarchy of groups of functions that represent subfunctions that can be grouped together into one major function. So using these kind of things like the Bingo plugin, which is a Sinoscape plugin, can help identify these kinds of relationships and give you more confidence that this particular function, this particular category really is enriched in this particular group of samples. All right, so in the lab later after this lecture, we are going to be taking you, Tony is going to be taking you through the steps of differential expression analysis using DESeq2, and then also taking you through the steps of gene set enrichment analyses as well. But the next thing I want to talk about is topic modeling. So before I move on to topic modeling, does anyone have any comments about these kind of statistical analyses using these tools? Yes.
I just wondered, are there a lot of gene sets really available? Because you gave it AMR and but are there some or methanogenesis? Or I don't think a bunch of them.
No, I think yeah. So what we try and do is, as we're analyzing a new metatranscriptomics data set, we have gene sets that we've curated or other people have curated that we include in our analyses. So we use these protein interaction data sets for cell wall biogenesis, for example. We have FE-Genie for the iron stuff. We have one for biofilm machinery now. There's a CARD database for antimicrobial resistance genes. So we're trying to feed in more and more of these modules to build up more of a consortium. But it's, again, this goes back to this lack of this resource for categorizing bacterial genes into these kind of functional modules. And again, I think it's a real limitation. So if there is something for methanogenesis, presumably that could be covered by metabolism. And there's probably some methanogenesis kind of pathways that would capture that kind of functionality. But if you're looking at like transporter machinery and those kinds of things, right, you've kind of got to assemble those yourself. So it depends on what you're interested in looking at. Is there a gene set, or do I need to curate my own gene set to address exactly what I'm interested in and the questions that I'm asking in my analysis? But if we are doing an analysis for the first time on trying to see what's different in this versus that, then you want to throw, and this maybe that goes against what Robyn was saying earlier, but you might want to throw a wide net to see which functions are differentially expressed between these data sets, because you're on a hypothesis generation exercise, right? And so you're trying to identify, is there some signal within all of this data? And then if there is something that looks interesting, then you can design follow-up experiments to actually validate whether that is really happening or not, right? So maybe we think of these kinds of analyses in these metatranscriptomic, metagenomic data sets as an initial hypothesis generation, then we do the specific experiment where we're targeting specifically those functions or those genes where we have predicted them from our initial experiment. Okay, good. Topic modeling. So what is topic modeling? So I mentioned that the idea here, it's a more nuanced version of coming up with enterotypes. It's trying to identify these kinds of sub-communities. That said, we have also applied this to enzymes, okay? So I think I mentioned this morning that KEGG pathways are a pretty good way of organizing how enzymes are functionally organized within the concept of a pathway. But these pathways on KEGG are curated based on metabolism, what's known about metabolism in humans, what's known about metabolism in yeast, what's known about metabolism in E. coli. Different organisms are going to have different routes for producing molecules, right? And so these kind of smaller modules or smaller interconnected regions of, you can think of metabolism as a big network, you're trying to identify sections in that network where there's some kind of coherence. And so topic modeling, you can apply in the same way as we have for taxa here, you can apply it to enzymes and see, are there groups of enzymes that have these kind of functional relationships which I haven't been able to, which I'm not able to capture using KEGG mapping, for example. So topic modeling, you can define communities. You can also define, if you like, you can think of them as sub-communities of enzymes, right? So, and that seems to work as well. So getting ahead of myself. So what is topic modeling? Well, it's this kind of approach where what you're doing is you're trying to convert and try and identify these so-called topics, these microbial sub-communities within your samples. And so each sample will be composed of several of these topics. And what these topics represent are collections of microbes. And a topic might be composed of, say, 60% of bifidobacterium, 30% lactobacillus, maybe 5% of Duria, right? So that's a representation of that topic. So you can think of, I guess, each of these topics as a kind of a sub-community with contributions from different taxa. And the nice thing about these topics is that different taxa can belong to more than one topic. Okay, so you're not just defining things into specific cliques. There's actually overlap between these cliques as well. And then there's something called latent Dirichlet allocation, LDA. This is the actual algorithm that is used to define these topics. Okay, so the idea here is, if you're thinking about your community, you have these taxa that are contributing to these different communities, and the traditional approach would be doing some kind of clustering, and you would allocate each of your taxon into a distinct cluster. And so each taxon is only associated with one clique. What topic modeling enables you to do is to say that a individual taxon can contribute to more than one community, and with a probability or a distribution which is defined by how much it's contributing to each of those different topics. Yeah. So there's an R package for this, fairly straightforward to run. The first step is to define K. Now, K is the number of topics that you're expecting to find in your sample, so i.e. the number of subcommunities. And the question is, how do you define K, right? Because you have to come up with that yourself. And so there's a method for doing that. It's based on this metric called perplexity, which is how well a probability model fits a new set of data. So you do this 5-fold cross-validation. Okay, so you split your data into 5, you remove one of those samples, you build your model based on those 4, and then you run your model against that 5th one that you left out and see how well it performs. And then using that, you calculate the perplexity, and you can end up identifying what your value of K should be. It should minimize K. So that's fairly straightforward. There's a method for doing that. Bish, bash, bosh, done. So all of this that I'm putting here is based on amplicon sequence data, but you could do it on enzyme data as well. We've tried doing it on gene data, but I think gene data, you'd have to collapse it down into kind of orthologs or something like that in order to come up with those kind of modules. We haven't tried that yet. But this is all based on amplicon data. Thank you for pointing that out. Next, you define the topics or the subcommunities, and then there's fairly straightforward R commands, which enables you to define those subcommunities. And then you end up with these lists of topics. And once you've got your list of topics, then you can apply something like DSeq2 or GLM to associate your topics with specific sample types. So here, for example, we have case versus control, and so we can see an upregulation of one of these topics. And then when we go and see what that topic is composed of, we can see that a major contributor is this Porphyromonaceae, right, which is that blob on the right-hand side. So you've kind of shrunk, or you've kind of collapsed all of your taxa into these topics, and then you can run your statistical significance tests, identify which topics are associated with one sample versus another sample. And then once you identify those significant topics, then you can go in and see what is that topic actually composed of. And in this case, a major contributor is this Porphyromonas, but there are other taxa associated with it as well. This is analysis that we did. It was a trial of 4 feed additives in poultry gut microbiomes. I've just got some, these are heat maps, and I've just circled a couple of topics that were downregulated in some of these communities. So we found that some sub-communities were decreased under treatment with yeast. Some of them were decreased under treatment with Avigard, which is this competitive exclusion product. So again, it's a way of us to identify what are the gross changes in this case, in the community associated with some of these treatments, and using topic modeling, it enables us to get a sense of how are these different treatments disrupting some of the sub-communities within these microbiomes. Okay, so any questions on topic modeling? I just thought it'd be a fun thing to throw out there for people to want to consider. It's a fairly new concept. I think people are only starting to apply this, but I think I like this idea that rather than focusing on specific taxa, we should be looking more at sub-communities, and that there's a lot of taxa that are functionally equivalent, and how do we capture that using these kind of approaches. And I think topic modeling, while not perfect, is potentially one route that we can do that. Tony.
Feel about using topic modeling, finding sex, running gene set enrichment. So it's a bit of a
Well, I think it comes down to the sweet spot of how many features or topic. Oh, what is the word I'm looking for? How many terms? There we go. So how many terms you have, right? So microbial taxa can represent one microbial taxon is one term, right? And so topic modeling relies on having these terms to describe, for example, the community. And I think the sweet spot is round about 100 to 500 terms. I think if you start getting more terms than that, so if you try doing it on genes, you just lose any signal. So if you're thinking about in terms of gene sets, I think it could be worth trying. If you think of KEGG pathways, there's what, 170 or 240 of them, depending on which version of KEGG that you're using. So that's potentially something that could be tried. I think the next step for my group is to try orthologs and try and collapse things into gene families and see if that also gives you some kind of signal as well. Yeah. Yes.
I guess it's being sort of an unsupervised clustering technique. Is it fair? And I guess if it's similar to like a self-organizing maps, I'm not sure if you're familiar with that. But it seems very similar.
It is a little bit of a clustering exercise, but it's creating these probabilistic models where each topic represents a probabilistic model that is composed of 30% of this, 60% of that, 10% of that, for example. So it's not like a clustering where you're specifically assigning one particular taxon into a group. That taxon could actually belong in multiple groups, or it's divided up amongst those groups. Yeah, I know that's not probably a very clear answer for you, but that's as good as I've got.
You do get a number of topics.
You do, so it's k-means clustering, right? So
Do you have to set it?
you have to set the k-means clustering ahead of time, yes. That's where the perplexity comes in, because that defines your k. Okay, good. All right, yes.
For the cost, like converted like to model analytics with US for the current big data. So for me, it's also kind of like it's a big model. And I think this part my understanding is like this one kind of each taxa can belong to different modules, different like topics. So it's nice because it could be potentially you affect both of them. You have multiple functions, but will it be less sparse? Like for example, I found 10 topics are interesting, and I open, And that, of course, the topics related to 100 different like species. When like I just wondering how sparse it is. Like for example, if some taxa only contribute 10%, maybe it shouldn't be there. It's just some things. Or a way to control that, or as long as it's over 50%, then I will add them. Always kind of like I feel like it's like you want to reduce dimension, but this one is kind of like reduce dimension, also increase dimension a bit. So just curious, yeah.
I think when you get these displays like on the right hand side here, you can see some of these taxa that are contributing, their contributions are so low that those are the ones that you probably discount, right? So when you start expanding what the topic is actually composed of, then that can give you those kind of insights. But I think the other thing it can do is, this isn't necessarily saying that porphyromonaceae is actually present in every one of those samples. It's saying that there's a 30% probability it's associated with those other taxa within that sample. But in another sample, you might have some of those other taxa that actually represent that kind of topic instead, if that makes sense. I don't know, it's a really interesting kind of idea that I think it gives us a new way of thinking about these kind of microbial communities, right? Rather than this particular taxon belongs with this subclass. It's kind of like, well, if you remove it and it's not there in this individual, these other two taxa are kind of replacing it instead. So it's getting at that kind of sense of what is kind of replacing what.
I think first let's look at what explains the different topics, as if it's cross-feeding or if it's phylogenetic relatedness and environmental filtering of some parts of the tree.
No. I think those would be super cool studies to do. Absolutely. Yeah. No, I think so there's only a couple of papers that have started applying it to microbiome, and they've just been using it as a way of doing the initial classification, but they haven't done any kind of the type of cool studies that you're talking about, like what are these things that are replacing each other? What is there, as you say, is there some kind of metabolic association with, you know, this topic is composed of these particular enzymes or whatever. So I think that'd be a great study to look at. Okay, it is 2:43. I'm going over. You know what? I've only got 2 slides, right?
Started a little bit.
And we did start a little bit late. Okay, and I blame Robyn because Robyn took, you know, an extra. He got no proof. Right. Anyway, there's only 2 slides, and I just want to touch on this concept of visualization of our microbiome data, right? So again, Ambio has these kind of cool tools to look at kind of genome structures and so forth. But if we're thinking about how do we get some kind of intuitive way to explore our data sets, yes, we can get these gene lists, we can get these gene ontology lists, we can do these bubble plots, we can do these bar charts. It all seems really unsatisfying because it's getting us away from the biology, right? And I'm more of a biologist, right? I'm interested in what are the functions, what are the bacteria actually doing? And so how do we visualize these kinds of functions? And so one approach, and this is one reason why we're doing this Cytoscape tutorial this afternoon. And Cytoscape is right at the end of the tutorial. I would suggest that you all make time, at least half an hour, to play around with Cytoscape. So skip to the end if you need to, because Cytoscape, it's just such a fantastically useful visualization tool that's interactive, enables you to explore your data. And it's basically allowing you to represent your data as a network. So you could use a heat map or you could use a network, and networks are way nicer to look at. In addition, within your network, you can have these nodes represent multiple kind of features. So here we've got a Cytoscape visualization. Each of these pie charts that you can see represents an enzyme that's involved in this metabolic pathway. And then each of the little bits of pie, each of the slices represents the taxonomic contribution to the expression of that enzyme, right? And so here you can see that the yellow, which is other bacteria, sorry, that's not very informative, should have chosen a better example. The green, there we go, the green is Clostridioides. So Clostridioides contributing a lot of expression within this particular part of the pathway, but Bacteroides, which is the purple one, you can see that that is a major contributor for some of the enzymes at the bottom of this pathway. So the details aren't important here, but the idea is that you've got a way of showing your set of enzymes and then looking at the taxonomic contributions to each of these enzymes. And these enzymes have connectivity, like there is functional meaning between the connections from one enzyme to another. They're sharing a substrate, right? So there's this substrate product relationship. So using these kind of views, you're getting more of a sense of which parts of this pathway maybe one taxon is contributing to. And so if you think about a very large map, so the one on the right-hand side is, I think, about 1,500 enzymes associated with it. You can zoom in using Cytoscape to identify parts of the network where you can see contributions to by individual taxa. So I think, you know, doing these kind of visualizations, I think the tutorial will only focus on one particular pathway, but Cytoscape, you can use it for visualizing all sorts. It doesn't have to be microbiome data, but you can think of it as a nice way of visualizing all sorts of different data that you can be generating in your analyses. And it's very intuitive. It's free. It's incredibly well supported. It's been going since 2012, I think it is, when the first iteration of Cytoscape came out, and they keep adding new functions to it. There's plugins. It's menu driven, so it's very intuitive. And so I just encourage everybody to kind of look at it, and maybe you'll find it useful for other types of analyses as well. So I really want to throw that out. And then the final thing I wanted to end up on is this is a really simplistic cartoony representation of this is the kind of view that I think we need to be moving towards in terms of interpreting these data sets. We have a microbiome, we are analyzing it, we're doing all of this crunching of this sequence data and coming out with which functions are present in our microbiome. How do we explore these different functions? Could we have something like a virtual bacterial cell where you can zoom in and point and click on different kind of sub-functions or different regions of the cell to look at flagellar expression, for example, or production of toxins or different metabolic pathways? And can we come up with these kind of resources as a community for bacteria? If we think of human data sets, for example, they have Reactome database, right, which is a really lovely database, but it's all focused on human kind of pathways and human resources. We need these kinds of data sets and resources for bacteria to help us visualize these kinds of relationships and give us a better way of exploring our data. And I think I'm going to stop there. Does anybody have any thoughts on this at all? We just wanted to throw this up more as a discussion point than anything else. What people think about, is visualization important to you, or are you just happy getting your lists of genes and gene categories? Yeah, it's again, as a biologist, it just seems so dissatisfying to get these lists, and you know, I really want to visualize these things.
I think there's kind of a duality. When I was talking to a few people today about this, bioinformatics, not to send Jekyll and Hyde when they're bioinformatician, you have to be a computer at some point, a robot that basically just is very analytical, making tables, making sure your stats are correct, making sure your workflow is correct, and you have to be kind of a creative, wild person, or a proper human, I suppose, that is able to look at things and look at the data in a creative way and start to think about how that affects the actual real world and put it in the context of the chaos that is biology. And I think that visualization is the part that is that part of our mind. And without that visualization, it's very hard to think creatively about your system. I really love tables. I love them. I love them to be clean. I love them. I like to see the stats. But if I want to try to, the more and more that I try to synthesize the data that I hold so dear, that I've spent so much time curating and making sure it fits in the hierarchy and so on and so forth, it's basically impossible to make an interesting story about biology without some kind of visualization. Interactive. There's almost none that are any good at all to do that. So, I like the last slide here. That I really wish we had that. It'd be awesome.
Yeah.
For anything, very communicate to other people. Like, this is in the little bacteria. This is happening.
Yeah, absolutely. All right. Great. So we are going to take a 15-minute. Wow, we're not getting any pushback on this. Oh, okay. Okay, so 20-minute break, and then we will dive into the last lab session. And as I said, so it will take you through DE-seq analysis. It will take you through gene set enrichment analysis. Then it will also describe how you can generate input from microbiome data to put into Cytoscape. And that'll be great to go back through, but I would really recommend to ensure that you do the very last. I mean, it's only a section about that long of the tutorial, but I would absolutely encourage you just play around. It's Friday afternoon. It's the end of a long week, and I think just clicking and moving things around, it's very therapeutic. I find it very calming. Like that. Where is that map? That map on the top right, I think that took me about 3 days to manipulate all of these nodes into pathways, into things that are in the same pathway. I found that very therapeutic at the time. So there's something about this kind of interaction where you're moving things around into something that looks and feels satisfying. Anyway, I really encourage you to spend time on Cytoscape. All right. Okay, let's have a break.